there are only 18 of you who are around. Okay? By the way, are you also the one that I'm going to lecture on the Krebs cycle at 4 o'clock this afternoon? Same group?
Let me check, sir.
I have another class at 4 o'clock.
Yes, sir. Try. Oh, no.
At 4 o'clock, right?
Yes, sir.
Krebs cycle.
Yes, sir. Try carboxylic acid pathway.
Yeah, okay. Okay, let me start my lecture now. The others will have to catch up with us. So this afternoon, we're going to discuss two metabolic pathways in carbohydrate metabolism. This will fall under the musculoskeletal module, and most of the topics in carbohydrate metabolism will fall under this MS module. So let us start our discussion of glycolysis and the pyruvate dehydrogenase reaction. First, let me describe to you the roles of carbohydrates in all forms of life. First and foremost, carbohydrates serve as energy store or metabolic fuel reserve, both in plants as well as animal tissues. Now, starch is the most abundant metabolic fuel reserve in plants, while glycogen is the metabolic fuel reserve in animal tissues. Principally, glycogen is stored in the liver and in the skeletal muscle. And tomorrow, we are going to discuss glycogen metabolism. Now, ribose and deoxyribose are pentoses. These sugars contain 5 carbon atoms in their molecule or in their structure. And they form part of the structural framework of DNA and RNA. So ribose in RNA and deoxyribose in DNA becomes associated with the nitrogenous bases, namely purines and pyrimidines. Now, the purines are the guanine and adenine, while the pyrimidines are cytosine, uracil, and thymine. And they become a very important component of the nucleus in the form of nucleic acids. Now, carbohydrates are structural elements in cell membranes of animal cells, as well as the cell walls of the bacteria and exoskeletons of arthropods. Now, cellulose is the main constituent of the plant cell wall. It is the fibrous framework in plants. And cellulose become an important part of our diet, especially when we eat vegetables and fruits. And we will discuss that also in some other modules. Now, carbohydrates become linked or become associated with proteins and lipids. Now, there are two kinds of carbohydrate containing proteins. They are glycoproteins and proteoglycans. The two differ only in the proportion of carbohydrates to proteins or vice versa. In proteoglycans, you have more carbohydrates than proteins. In proteoglycans, the carbohydrates are glycose aminoglycans. On the other hand, in glycoproteins, you have more proteins than carbohydrates. As a matter of fact, in glycoproteins, the carbohydrates are found only in the form of oligosaccharides. Oligosaccharides are carbohydrates containing 2 to 10 sugar units. You must have heard about chylomicrons, LDL, VLDL, HDL. They are hybrid molecules of carbohydrates and lipids. Okay, primarily phosphoglycerides. Now, these lipids are lipoprotein molecules. Okay? Now, carbohydrates can also generate reducing equivalents. And as you have studied in energy metabolism in the electron transport chain, carbohydrates can generate reducing equivalents in the form of NADPH, NADH, FAD, FADH2. And reducing equivalents are actually electrons that can be transferred from one substance to another substance. And this reduced NADPH, NADH, FAD, FADH2, they have important roles inside our body, particularly in ATP production and also in biosynthesis of important cellular components, especially NADPH. Now, why is glucose the most important carbohydrate? First, it is the characteristic sugar of blood and tissue fluids. It is as glucose that the bulk of dietary carbohydrate is absorbed into the bloodstream or into which it is converted in the liver. I think you already had a lecture on digestion and absorption of dietary carbohydrates by Dr. Ralph Jacinto. It is also from glucose that all other carbohydrates in the body can be formed. For example, glucose can be converted to glycogen, which, as I've said earlier, is the metabolic fuel reserve in our body, and glycogen is stored mainly in the liver and skeletal muscle. Now, all other structural components of the body in the cell membrane are derived from glucose, especially the glycose amino glycans. Now, glucose is a major fuel of the tissues of mammals and a universal fuel of the fetus. That means to say that the major source of energy in our body wait, notification appeared in my screen. It's okay. I was given unlimited minutes for this lecture. What am I saying? I'm saying that glucose is the major source of energy inside our body. The metabolism of many proteins and amino acids proceeds by glucose pathway, and some products of glucose metabolism can be utilized for the biosynthesis of some amino acids. And I will illustrate that as I go along. Now, what is the first reaction that usually happens to glucose once it is already inside the cell? Glucose becomes phosphorylated to glucose 6-phosphate. And glucose 6-phosphate is a central metabolite in the metabolism of glucose and other carbohydrates like fructose and galactose. The reaction is catalyzed by either one of these two enzymes, hexokinase and glucokinase. Now, glucokinase is an isomeric form of hexokinase, and please take note that kinases in general are phosphorylating enzymes that derives the energy from ATP, and the phosphate group in ATP is transferred to the substrate of hexokinase and glucokinase in order to form glucose 6-phosphate. Now, let us differentiate hexokinase from glucokinase, and this is very important. Now, hexokinase has a low Km for glucose. On the other hand, glucokinase has a high Km for glucose. Okay? Now, you have learned in your lecture on enzyme chemistry that Km, the so-called Michaelis constant, reflects the chemical affinity of the enzyme for its substrate. In this particular case, it reflects the Km of the enzyme reflects the chemical affinity of the hexose as well as glucokinase for its substrate, which is glucose. And what is the relationship? It is inversely proportional. A low Km means that the enzyme has a high chemical affinity for glucose. On the other hand, a high Km means that glucokinase has a low chemical affinity for glucose. Okay? And what is the repercussion of that? That means to say that hexokinase, since it has high chemical affinity for glucose, needs only a small amount of glucose to reach 1/2 maximal velocity of the enzyme catalyzed reaction. In other words, 1/2 Tmax. I'm sure that we have discussed this in enzyme chemistry. Glucokinase, will you please mute? Will you please mute your microphone because it is interfering in my lecture. Come on, please mute your mic.
అరే బ్లూటూత్ ఇస్తావా
Apalanga. Mute your microphone, please. In other words, glucokinase, which is a low Km for glucose, needs a higher amount of glucose in order to achieve 1/2 Vmax. Okay? So 1/2 maximal velocity of the enzyme catalyzed reaction. The role of hexokinase, therefore, is to maintain a normal tissue level of glucose at normal glucose blood concentration and even at lower than normal blood glucose level. On the other hand, glucokinase, its role is to bring down the elevated blood glucose level that usually happen after eating your meals. Okay? After eating your meals. So I want you to review your enzyme chemistry when it comes to enzyme kinetics, the Km of the enzyme, and the reaction velocity. Now, there are other differences, and they are the following. Now, hexokinase is inhibited by high level of glucose 6-phosphate, while glucokinase is not inhibited by the end product of the reaction. Hexokinase is non-specific. It can catalyze other phosphorylation reaction involving other sugars like fructose and mannose. Well, actually, this is what we call relative specificity. Because what is the relationship of glucose with fructose and mannose? Well, first of all, they are sugars and they belong to the same group. They are hexoses containing 6 carbon atoms. On the other hand, glucokinase acts only on glucose. Hexokinase is well distributed in all tissues of the body except the liver, while glucokinase is found in the liver and in the pancreas. Okay? Now, I mentioned earlier that glucose 6-phosphate is a central metabolite in the metabolism of glucose. So these 7 are the metabolic fates of glucose 6-phosphate. Okay? You can remove the phosphate group in carbon 6 of glucose and you convert it back to free glucose. And this free glucose, with the help of glucose transporters, can diffuse out of the cell into the circulation. It can enter glycolysis, which is our main topic this afternoon. And from glycolysis, the pyruvic acid, which is its end product, will be acted upon by pyruvate dehydrogenase enzyme complex, which is also our topic this afternoon, ending with the formation of acetyl coenzyme A. And acetyl coenzyme A, together with oxaloacetate, are the first reactants in the Krebs cycle. And glucose will first undergo oxidative degradation to carbon dioxide at the end of the Krebs cycle. Glucose 6-phosphate can be used for the biosynthesis of glycogen and stored in the muscle and in the liver. Now, there is an alternative pathway for the oxidation of glucose 6-phosphate, and that is via the hexose monophosphate shunt, also called pentose phosphate pathway. Now, what is the importance of the uronic acid pathway in plants and in lower animals? In the uronic acid pathway, glucose 6 phosphate can be converted to ascorbic acid or vitamin C. Unfortunately, we humans and other primates cannot do that. We cannot convert glucose to vitamin C because there is an enzyme that is deficient. We'll have another lecture on that. Now, glucose 6 phosphate, when converted to acetyl CoA, can be used for the synthesis of fatty acid as well as cholesterol. We're going to take that up in detail in lipid metabolism. So let us now discuss glycolysis. It is the major pathway for glucose utilization, and it is found in all cells of the body. And it sets the stage for the complete oxidation of glucose to water, carbon dioxide, and the concomitant formation of ATP via the pyruvate hydrogenase reaction followed by the Krebs cycle. Now, the Krebs cycle is linked to the electron transport chain, and the electron transport chain is in turn linked to oxidative phosphorylation. So most of the ATPs inside our body are generated through oxidative phosphorylation. The reducing equivalents, okay? The reducing equivalents that will transfer electrons to the respiratory chain and coupled to oxidative phosphorylation comes from glycolysis as well as the Krebs cycle. Now, glycolysis is a unique pathway since it can utilize oxygen if available, and we call that aerobic glycolysis, or it can function in the absence of oxygen, and that is anaerobic glycolysis. Okay? So glycolysis is a sequence of reaction that converts glucose to pyruvic acid. That is the end product of aerobic glycolysis. Or it may end up as lactate when it is anaerobic glycolysis. Either way, we produce ATP. But we would rather that we have aerobic glycolysis because we produce much more ATP than when we use anaerobic glycolysis. And we're going to illustrate that later on when we discuss the bioenergetics of beta or of glycolysis. And later this afternoon when we talk about the Krebs cycle. Now, all of the enzymes involved in glycolysis are present in the cytosol. So that is the cellular localization of glycolysis. Glycolysis is basically an exergonic process that releases energy, and this energy is ultimately trapped in the form of a high energy compound called adenosine triphosphate. Glycolysis also provides the main pathway for the metabolism of fructose and galactose. We get fructose and galactose from our diet, and their metabolism proceeds through the metabolism of glucose. Now, all intermediates between glucose and pyruvate are phosphorylated. Why so? Because of the action of hexokinase and glucokinase. kinase that initiates the conversion of glucose to glucose 6 phosphate. So all other intermediates, therefore, are phosphorylated. Now, what are the functions of the phosphate group in the structure of the intermediate metabolites of glycolysis? Number one, the phosphate groups serve as binding or recognition groups in the formation of the enzyme substrate complex. Okay? So in other words, the enzyme must be able to recognize its substrate. And in this particular case, it is the phosphate group that is recognized by the enzyme. And they provide each intermediate with a polar negatively charged group. And what is the importance of this number 2 item here? This means to say that the intermediate metabolites cannot go out of the cell anymore, because as you have learned in cell membrane, the lipid bilayer will not allow polar and so much more charged particles to pass through it. So once the intermediate metabolites become phosphorylated, there is no way by which they can go out of the cell. They have to go forward to the subsequent reactions of the pathway. Okay? And third, the phosphate groups function in the conservation of energy, simply because this phosphate group becomes the usually the third phosphate group in adenosine triphosphate. Okay? Not only in the course of glycolysis, but also in the other pathways of carbohydrate metabolism. So please remember these three functions of the phosphate group in the intermediate metabolites of glycolysis. So what are the functions of glycolysis? Well, I said that already before, repeatedly, to produce energy in the form of ATP. Number two, intermediates formed can be converted to other substances like amino acids and fatty acids and other substances. For example, what did I say about the end product of aerobic glycolysis? Will you please answer me, class?
Pyruvate.
Very good. Pyruvic acid. And when pyruvic acid undergo transamination reaction, it can be converted to alanine, an amino acid. Okay? Now, these intermediates can proceed through the formation of acetyl-CoA. And acetyl-CoA, as I've said already, can be used as a substrate in lipogenesis, which is fatty acid synthesis, as well as cholesterologenesis, the synthesis of cholesterol. Now, these are the major organs and tissues of the body that utilizes glycolysis as their major mechanism for ATP production. By and large, what do they have in common? They either lack mitochondria, or if they have mitochondria, there are only very few mitochondria inside the cell. And this is particularly true of the mature red blood cell, the cornea, the lens of the eye, regions of the retina. They do not have the mitochondria, and therefore they do not have the Krebs cycle. They do not have the electron transport chain. They do not have oxidative phosphorylation. So the energy comes from the glycolytic pathway. Now, the kidney medulla, the renal medulla, that's the inner portion of the kidney, the testes, the leukocytes, white muscle fibers, they are almost totally dependent on glycolysis as a source of ATP. And this is what I'm saying. They have few mitochondria. So combine the tissues that are dependent primarily on glycolysis for ATP production consume around 40 grams of glucose per day in the normal human adult. Now, one of the great users of glucose and of the glycolytic pathway is the central nervous system, particularly the brain. It has an absolute need for glucose and processes most of it via glycolysis. Approximately, look at the number. 120 grams of glucose is used by the adult human brain each day in order to meet its extraordinary need for ATP. I don't think I have to overemphasize on that. Okay? Now, the heart muscle is adapted for aerobic performance and has relatively poor glycolytic ability and therefore poor survival under conditions of ischemia. What do you mean by ischemia? Lack of blood supply, okay? Lack of blood circulation into the affected area, okay? Lack of blood supply, lack of oxygen, lack of nutrients, and the muscle can be infarcted, and the person will suffer from myocardial infarction or heart attack. Now, look at this. In fast-growing cancer cells, glycolysis proceeds at a much higher rate than is required by the Krebs cycle. So in other words, there will be a backlog of pyruvate. Okay? So more pyruvate are produced that can be metabolized via the pyruvate dehydrogenase reaction and the Krebs cycle. And what will happen to this pyruvic acid? It will be converted to lactic acid. And elevated levels of lactic acid will lead to lactic acidosis. So there will be pyruvate acidosis and lactate acidosis. And what kind of acid-base imbalance is this? It is metabolic acidosis. The normal pH of the body is 7.4 plus or minus 0.05. Once you go down below 7.35, you will already be in a state of acidosis, and that can kill you. Okay? Now, there are two phases of glycolysis. The energy investment phase, also called priming states, wherein ATP is used. Okay? Now, this starts from glucose up to the formation of fructose 1,6 bisphosphate. Okay, let me clarify this. Is it correct to say diphosphate, fructose diphosphate, or is it better termed bisphosphate? Well, you see, before we do not mind at all, okay? Because there are two phosphate groups attached to carbon 1 and attached to carbon 6. Now, usually, when do we call it diphosphate or triphosphate? Example, ATP, ADP, adenosine diphosphate, adenosine triphosphate. You do not hear us or anybody saying adenosine bisphosphate, adenosine trisphosphate. Now, you use diphosphate, triphosphate when the phosphate groups are attached to one another and a single phosphate group becomes attached to a carbon atom. Okay? So, AMP, adenosine monophosphate, attached to uh, attached to the 6 carbon atom, for example. Okay? Or first carbon atom. Okay? And then the second is attached to the first phosphate group. The third phosphate group is attached to the same or to the preceding phosphate group. Okay? But here, when the phosphate groups are attached to different atoms, carbon atoms, you rather call it bisphosphate. Okay? So, one phosphate is attached to carbon 1 of fructose, the other to carbon 6 of fructose. Second, energy generation phase, also called energy recovery phase, wherein ATP is generated. And there are two reactions involved here, the splitting states. And it is very important to remember this. Very, very important. Why? In the splitting states, the 6 carbon atom fructose, a hexose, the 6 carbon atom fructose is cleaved into half. Okay? It is cleaved into two trioses, 3 carbon atom sugars, namely dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Okay? Because after the splitting stage, you count by twos already. Because ultimately, the dihydroxyacetone phosphate will be converted to glyceraldehyde 3-phosphate. So now you metabolize further or you degrade further two molecules of glyceraldehyde. Okay? We'll illustrate that in an illustration. Another part of this energy generation phase is the oxidoreduction phosphorylation stage, and it extends from glyceraldehyde 3-phosphate down to the formation of either pyruvic acid or lactic acid. Okay, remember these three reactions because you are going to encounter this again when we discuss gluconeogenesis. Gluconeogenesis. These are the three irreversible steps in glycolysis. And in gluconeogenesis, you have to bypass these three irreversible steps using another enzyme. Okay? Using another enzyme. Because what is gluconeogenesis? It is the biosynthesis of glucose from non-carbohydrate precursors. And it is an adaptation of the Krebs cycle and the glycolytic pathway. We'll discuss that in gluconeogenesis. What are these three reversible steps? Two of them are in the energy investment phase. The first one is a conversion of glucose to glucose 6-phosphate catalyzed by any one of these two enzymes, which we have discussed already and differentiated the two from each other. And the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, wherein you add another phosphate group, that is to carbon 1 of fructose. And what is the significance of this step? It is the rate limiting step catalyzed by phosphofructokinase 1, which is the committed enzyme. And the third irreversible step is the conversion of phosphoenol pyruvate to pyruvic acid catalyzed by pyruvate kinase. So remember the reactions, remember the enzymes involved. Okay, now these are the other names of the glycolytic pathway. Okay, and then Meyerhoff pathway. Okay, and then Meyerhoff Parnas pathway. Okay, so let us summarize what we have mentioned before. Glucose catalyzed by hexokinase or glucokinase will be converted to glucose 6-phosphate, and it will utilize 1 ATP, which is hydrolyzed to ADP plus inorganic phosphate. And the inorganic phosphate will become attached to carbon 6 of glucose. Then there will be an isomerization of an aldohexose. It contains an aldehyde group to a ketohexose containing a ketone group. And that is the conversion of glucose 6-phosphate to fructose 6-phosphate. Glucose is an aldohexose, while fructose is a ketohexose. The reaction is reversible, catalyzed by phosphoglucose isomerase. Now, the other ATP is used by phosphofructokinase 1 to convert fructose 6-phosphate to fructose 1,6-bisphosphate. Okay? And that is the end of the line for the energy investment phase. And from here on, it is the energy recovery phase, wherein first, fructose is acted upon by aldolase. So fructose is split into two products, two trioses, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And take note, okay, the reactions are reversible, okay, reversible reaction. And look at this one. Phosphotriose isomerase converts DHAP, that one, to glyceraldehyde 3-phosphate, GAP, GAP, glyceraldehyde 3-phosphate. It is glyceraldehyde 3-phosphate is in the direct pathway of glycolysis. Now, let us let us go to a corollary matter, a corollary topic. Look at that. That is phosphofructokinase 1. Okay? There is an isoenzyme called fructose 6 phosphofructokinase 2. Phosphofructokinase 2. It converts fructose 6-phosphate to fructose 2,6-bisphosphate. Take note of that. PFK1 converts fructose 6-phosphate to fructose 1,6-bisphosphate, while PFK2 converts fructose 6-phosphate to fructose 2,6-bisphosphate. And what is the significance of this product? Okay, fructose 2,6-bisphosphate activates phosphofructokinase 1, which is the rate-limiting step in glycolysis, thereby stimulating glycolysis. If you stimulate glycolysis, you stimulate utilization of glucose by all cells of the body, because glycolysis is found in all cells of the body, and therefore that will decrease the blood glucose level. Another thing, fructose 2,6-bisphosphate inhibits fructose 1,6-bisphosphatase, okay? A regulatory enzyme in gluconeogenesis. So by inhibiting bisphosphatase, you inhibit gluconeogenesis. You inhibit the formation of new glucose. Okay? And what is the overall effect? Decrease fructose phosphofructokinase 2 through fructose 2,6-bisphosphate decreases the blood glucose level. Okay? Decreases the blood glucose level. Okay, let's continue the energy investment phase. What happens to glyceraldehyde 3-phosphate? It will undergo dehydrogenation reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase. You extract electrons from glyceraldehyde, transfer it to oxidized NAD, thereby reducing NAD to NADH. Okay? When under aerobic condition, when there is oxygen, this NADH will be translocated into the mitochondria, and it will enter the electron transport chain at complex 1 of the respiratory chain, which you have learned already before. Okay? And when that happens, what is the PO ratio? 2.5. Okay? Now, that converts glyceraldehyde 3-phosphate to 1,3-diphosphoglycerate. Okay? And there will be substrate level phosphorylation here, catalyzed by phosphoglycerate kinase. Okay? The phosphate group in carbon 1 will be removed by the kinase, transferred to ADP, and ADP becomes ATP. And 1,3-diphosphoglycerate, which is a high energy compound, will be converted to a lower energy phosphorylated compound called 3-phosphoglycerate. So what do we have here? ATP production through oxidative phosphorylation. And what do we have here? ATP production through substrate level phosphorylation. The direct transfer of a phosphate group from a high energy compound 1,3-DPG to ADP. So what happens next? 3-phosphoglycerate will be acted upon by phosphoglyceromutase, and it is converted to 2-phosphoglycerate. Okay? Mutase is an isomerase enzyme. There are different kinds of isomerases, of which mutase is one of them. Okay, next. 2-phosphoglycerate, okay, will be converted to phosphoenol pyruvate, okay, catalyzed by enolase. There will be a dehydration reaction. And then phosphoenol pyruvate, another high energy compound, okay, will be acted upon by pyruvate kinase. And the phosphate group will be transferred to ADP to form ATP. And that is another substrate level phosphorylation step, forming pyruvate. Okay, now take note of this. In every substrate level phosphorylation step, you only produce one ATP at a time. But look at this one. Let me go back to this one. Isn't it that fructose is converted to 2-trioses? Okay? And that dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. So from here on, there are two glyceraldehyde 3-phosphate molecules that you degrade in the energy recovery phase. So what happens? You produce 2 NADH. So 2 NADH will enter the electron transport chain. Okay? The PO ratio is 2.5 times 2. That's a total of 5 moles of ATP. Here, okay? How many 1,3-diphosphoglycerate will undergo substrate level phosphorylation reaction? 2 of them. So 2 ATPs. Same here, 2 ATPs. So when it is aerobic glycolysis, you end up here with pyruvate, with pyruvic acid. But if it is anaerobic glycolysis, pyruvate will be converted to lactate, catalyzed by lactate dehydrogenase. The reaction is also reversible. The conversion of pyruvate to lactate is oxidation, is reduction reaction, okay, wherein NADH will transfer electrons to pyruvate, so pyruvate is reduced to lactate. By the way, where did we get this NADH? We got it there in the glyceraldehyde 3-phosphate dehydrogenase. Remember, anaerobic. So this NADH cannot go to aerobic glycolysis. Okay? It will not go to aerobic glycolysis. So it will be used to convert pyruvate to lactate. And in the reverse reaction, NAD, lactate will be oxidized to pyruvate. Lactate will transfer electrons to NAD. NAD will be reduced to NADH, and lactate becomes pyruvate. Okay? Now let us discuss this. That one. How do we translocate NADH? How do we translocate NADH to I'm drinking water. How do we translocate NADH to the mitochondria? We use two shuttle mechanisms. The first one is the malate aspartate shuttle. Where is that NADH? There. That is the product of the glyceraldehyde 3-phosphate reaction. It will be acted upon by malate, cytosolic malate dehydrogenase, because that NADH is generated in the cytoplasm where you have glycolysis. It will react with oxaloacetate. NADH will transfer the electrons to oxaloacetate, thereby reducing oxaloacetate to malate. And in turn, NADH becomes oxidized to NAD. Then, using the malate aspartate shuttle, malate will pass through the inner mitochondrial membrane using an alpha-ketoglutarate transporter. Number 1, alpha-ketoglutarate transporter. And so malate is now in the mitochondria. It will react with oxidized NAD in the presence of malate, mitochondrial, malate dehydrogenase. Look at that. There are two such enzymes, one present in the cytosol, the other present in the mitochondria. And so what will happen? Malate will transfer electrons to NAD. So what does malate contain? Malate contain electrons coming from this NADH over here. And then in the mitochondria, the electrons contained in malate will be transferred to NAD. And NAD is reduced to NADH, and that will go to the electron transport chain, whereas in protein complex 1. Okay? And malate will be converted back to oxaloacetate. And then what will happen to this oxaloacetate? It will undergo transamination reaction catalyzed by transaminase enzyme. It will react with glutamic acid. So oxaloacetate will be converted to alpha-ketoglutarate, and alpha-ketoglutarate will be translocated into the cytosol by alpha-ketoglutarate transporter. And alpha-ketoglutarate will react with aspartic acid. So in another transamination reaction, alpha-ketoglutarate plus aspartate, alpha-ketoglutarate will become oxaloacetate, aspartate will become glutamate. Okay? I repeat, alpha-ketoglutarate will become glutamate while it is aspartic. I made a mistake there. Alpha-ketoglutarate will undergo transamination reaction with aspartic acid. So, in so doing, alpha-ketoglutarate will be converted to glutamate, while aspartic acid will be converted to oxaloacetate. Okay? Here, oxaloacetate will become aspartic acid, while glutamate will become alpha-ketoglutarate. Okay? Let me correct myself there. And this malate aspartate shuttle is used by the kidney, by the liver, okay, to transport electrons from the cytosol to the mitochondria. Okay, so you have a number 2 enzyme transporter here, glutamate aspartate transporter. Okay. Now, another shuttle mechanism which is primarily used by the skeletal muscle, and this is the glycerol phosphate shuttle. This is the NADH that we generated, okay, in the glyceraldehyde 3-phosphate dehydrogenase reaction. It will react with dihydroxyacetone phosphate, okay, catalyzed by cytosolic glycerol 3-phosphate dehydrogenase. NADH will transfer electrons to dihydroxyacetone phosphate. NADH will be reoxidized back to NAD. Dihydroxyacetone phosphate will be converted to glycerol 3-phosphate. So the electrons formerly carried by NADH will now be carried by glycerol 3-phosphate and it will be translocated into the mitochondria, where it will be acted upon by glycerol 3 phosphate dehydrogenase in the mitochondria. It will react with fat, and so what will happen? Glycerol 3 phosphate will be converted back to dihydroxyacetone phosphate, which will diffuse back to the cytosol, while the electrons carried by glycerol 3 phosphate will be transferred to fat, forming FADH2. And FADH2 will go to the electron transport chain. At what protein complex? Complex 2. Okay? And what is the PO ratio for FADH2? 1.5. Okay? So this is the what do you call that? This is the bioenergetics of glycolysis only. Okay? So, pathway involved, glycolysis, reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase, method of energy production, respiratory chain oxidation of 2 NADH, 2.5 times 2, 5. That's the PO ratio for NADH. Then you have 2 substrate level phosphorylation. Okay? And you produce 2 there and 2 there. Okay? And that will give you 9. A gross of 9 molecules of ATP. You have to subtract the 2 ATPs that you use in the what do you call that? In the energy investment phase. Okay? So the net ATP production for glycolysis only is 7. And that is for every glucose molecule. For every glucose molecule. Now, for the pyruvate dehydrogenase reaction that we're going to discuss. Oh, by the way, okay, since you're the same group of first year that I'm going to lecture at 4 o'clock, okay, I think it is much better for me to discuss the pyruvate dehydrogenase reaction together with the Krebs cycle. But remember this, in the examination, when I make the examination, I will make the questions from pyruvate dehydrogenase reaction, I will make it together with glycolysis. Okay? So I was asked to by Dr. Maniwa to prepare 20 questions. And I'm going to prepare 20 questions both for the glycolysis and pyruvate dehydrogenase reaction combined. Okay? And a separate 20 questions for our lecture at 4 o'clock. And that is for the Krebs cycle. So let us finish this. Enzymatic control of glycolysis, phosphofructokinase. Remember this, okay? This is what is quite difficult for many students. But the general rule is this. When the cell is already fully charged with energy, you inhibit glycolysis. You inhibit the pyruvate dehydrogenase reaction. You inhibit the Krebs cycle. Now, what is daunting to the students is, how will I know that the cell is already charged, fully charged with energy? Well, look at this one. Phosphofructokinase 1, the rate-limiting enzyme, is inhibited by ATP, citrate, hydrogen ion that is present in NADH plus H, and cyclic AMP, and increased ATP AMP ratio. Remember all these. Okay? When you have high levels of ATP, you have high levels of citrate, which is the first intermediate product in the first reaction of the Krebs cycle, when you have plenty of hydrogen ions from NADH plus H plus cyclic AMP, and you have an increased ATP AMP ratio, that means to say that you have high ATP, low AMP. That means to say that the cell is charged with energy, and that will inhibit, in general, phosphofructokinase, hexokinase, pyruvate kinase. Okay? Now, on the other hand, how will you know that the cell is undercharged of energy when there is plenty of ADP, AMP, which are the byproducts of ATP hydrolysis? Decreased ATP AMP ratio means to say you have high AMP and lower ATP. Okay? Although you can still have higher ATP but AMP, but in general you have higher AMP compared to ATP before. Okay? Decreased ATP AMP ratio. Another thing, glucose 6-phosphate is an inhibitor of hexokinase but not glucokinase. And pyruvate kinase, inhibited by ATP, alanine, fatty acids, acetyl-CoA. Why alanine? What did I say earlier? That when you have an excess of pyruvate, you convert it to alanine through transamination reaction. Okay? And stimulated by fructose 1,6-bis phosphate. Now, there are other inhibitors of the glycolytic pathway. I just require you to know what they are and what enzyme is inhibited. 2-deoxyglucose inhibits hexokinase. Sulfhydryl reagents like iodoacetate inhibits glyceraldehyde 3-phosphate dehydrogenase. Fluoride inhibits inulase. Okay? So, know what enzyme is inhibited. Okay? And this one will be discussed this afternoon. Oh, I'll start it. Phage of pyruvic acid, reversible reduction to lactate, catalyzed by lactate dehydrogenase. Okay? So, how do you convert lactate to pyruvate? Through oxidation reaction. Okay? Lactate becomes oxidized to pyruvate, catalyzed by lactate dehydrogenase using NAD as a coenzyme. Conversion back to glucose. How? Gluconeogenesis. By bypassing the irreversible steps in glycolysis. You will notice, as I've shown before, that most of the regulation of glycolysis occur at the level of the irreversible steps of glycolysis. Formation of oxaloacetate or malate. Transamination to form alanine. Okay? To form alanine. This one, formation of oxaloacetate or malate, that is through what? Transamination reaction also, like this one. And this is the one that we are going to discuss at 4 o'clock together with the Krebs cycle, because the discussion of the Krebs cycle is not that long. Okay? So, we're going to discuss this. Oxidative decarboxylation of pyruvic acid to form acetyl coenzyme A. Okay? So, it's 3:10. Okay? I'm going to see you at around 4 o'clock. Okay, class? Let's have a snack break. Okay? Are you there? Are you still listening?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Sir.
Already. Bye-bye for now.
Bye, sir.